Hi, my name is Andrew Curd, and this is my Unit 4 presentation on the review article Gene Therapy Coming of Age. So, genetic therapy, in contrast to protein based drugs, is that while protein based drugs may require repeated infusion, gene based therapies are aimed to create long lived cells that might afford sustained protection for the host. And they aim to do this in maybe one or just a few amount of treatments as opposed to the constant treatment that other drugs may require for these same types of genetic conditions. So, translation of gene therapy concepts to patient care began in the early 1990s, but um, this was plagued by disappointing clinical trial results because of the emergence of serious toxicities related to the gene transfer efficiency, which resulted in some patient deaths. However, over the recent years, further maturation of the science of gene therapy, safety modifications, and improvements in gene transfer efficiency have finally delivered um, more clinical progress. One of the ways that gene therapy is used is with viral vectors. The most common viral vectors are retrovirus and adeno-associated virus. Um, however, these type of viral vectors can only mediate one type of gene modification, which is gene addition, and they do have the potential for genotoxicity with off-target editing. However, viral vectors aren't the only way that genetic editing can happen. There are many other forms, such as usually zinc finger nucleases or CRISPR-Cas9, and using these other forms can do not only gene addition, but also gene ablation, gene correction, deletions, and other genomic modifications to the cell. Um, using these more specific genome editing, such as CRISPR-Cas9, they overcome a lot of the drawbacks of the viral vector, as there should not be any genotoxicity because CRISPR-Cas9 is so on target with its editing. There won't be very much off-target editing, but these do come with their own drawbacks, such as getting the all of the editing components into the target cells. But genome editing as a therapeutic modality is already showing up in the clinic, as there have been T cells that have been edited to be less likely to cause graft versus host disease by deleting the T cell receptor gene. Um, and even more recently is in China, they edited non-viable human embryos. However, this editing of human embryos has sparked a lot of concern due to the ethical considerations of editing the human fetuses. And this sparked concern all around the world and created an advocate group for the governance of human genome editing, just to deal with these specific ethical considerations. Um, So there are a couple ways that genome editing is done. It can be done in vivo or ex vivo. So doing it in vivo is attractive because it avoids the practical regulatory hurdles of ex vivo cell-based gene therapies, because cell-based gene therapies require cell collection, culture, manipulation, and transplantation. While doing it in vivo is you just have to dose the patient, and it would go to the cells they already have in their body. However, the problems with this come from being on target or getting the cells to the proper place. I mean, not the cells, the editing components to the proper location in the body. Um, or, ex vivo is still used a lot due to the fact that doing ex vivo means that you can use the patient's own cells, edit them, and put them back in their body. So there shouldn't be any toxicity or rejection of these cells. And one of the ways that ex vivo is being used today is with CAR therapy. So T cells are engineered for a specific receptors and antigen, which reprogram the T cells so that they can activate themselves and help fight whatever they are programmed to fight. And this is used for the treatment of certain cancers. And it has also been used for other autoimmune disease disorders and AIDS. So, while many advances have been made in the for gene editing and gene therapy, many challenges remain, and these include addressing the genotoxicity from integrating gene delivery vectors or off-target genome editing, also improving the gene transfer editing efficiency to levels necessary for effective treatment, and one of the main hurdles that they'll have to come is reaching a social consensus regarding the ethical issues such as germline editing, and also for payment of expensive curative therapies. Overall, I really enjoyed this review article. I think it highlighted a lot of the key components of genetic editing and the advances that we've made in the past years for them, and it paints a bright picture for the future. However, this article was written in 2018, and I would be excited to see what the past 2 years have brought to genetic editing, as I know that the research for this has never stopped. That's my presentation. Thank you.